So we have seen that small substances are able to use passive or active transport proteins and transporter channels in order to enter or leave the cell. Larger substances like hormones, proteins, certain amino acids, cellular debris and wastes, and things like this that are relatively larger than the sugars and the small ions that we dealt with when we were talking about diffusion and active transport, these larger substances have to use a different mechanism to enter or exit the cell. And in order to do this, to move these relatively large substances and material, the cell makes use of these bubble-like sacs that are called vesicles to transport these relatively large materials. Now, vesicle transport is a form of active transport as it makes use of ATP energy to drive it. Although we won't go into the specific mechanism and details of exactly how that works, just know that vesicle transport is a form of active transport for the cell. So the cell has to invest cellular energy, typically in the form of ATP, in order to power vesicle transport. So vesicle transport can be divided into two categories. So that is, if material is entering the cell, that's referred to, or if material is entering the cell by vesicle transport, we refer to that as endocytosis. And materials, large materials that are exiting the cell by vesicle transport is referred to as exocytosis. So we have endocytosis going into the cell and exocytosis, materials being ejected to the external environment. So let's take a look first at endocytosis, because there's really only one form of exocytosis, but there's a couple of different types of endocytosis, and we'll look at where those are used in the body and for what specific purposes. So focusing on the three mechanisms of endocytosis, we see that there is phagocytosis, pinocytosis, and receptor-mediated endocytosis. So we'll start with phagocytosis first. So in phagocytosis, we have a relatively large piece of material that binds to receptors on the plasma membrane here. And so this blue structure that we see here, this blue wavy structure, that's our plasma membrane. These purple Y-shaped structures that we see are the receptors for this particular substance or piece of material that's outside of the cell here. So we have a relatively large piece of material binding to receptors on the outside of our cell, and in response to this, the cell is going to extend its plasma membrane around the material. So it essentially surrounds the material with a little extension of plasma membrane and encloses it in a vesicle and brings that material into the cell here. Now, typically this material that's in the vesicle is combined with a digestive organelle that's called a lysosome, and the lysosomes contain digestive enzymes. And typically what happens is that when this vesicle combines with that lysosome, those enzymes are released from the lysosome into the vesicle, and the material that's within the vesicle tends to be digested or broken down in some way or form. At the chemical level, I should say. So this process, as you probably imagined, is very highly selective because the material that we're bringing into the cell has to bind to specific receptors on the cell surface. So if that specific material is not present and it doesn't bind to the receptors, the cell is not going to bring anything in. And so really the receptors will only bring material in when that material is present. Phagocytosis is a pretty common mechanism that's used by cells of the immune system, particularly cells of the immune system whose function it is to destroy bacteria, foreign microorganisms like fungi, yeasts and things like that, or even dead tissues or parts of dead cells or things like that. So phagocytosis is really used by the cleanup crew, if you will, of the immune system, that those cells that are removing potentially harmful microorganisms or material from dead tissues and cells. Now, in pinocytosis, we see that the cell creates a little bubble or a little vesicle in its plasma membrane, and that vesicle fills with material and fluid from the outside of the cell. And then what happens is the cell fully encloses this vesicle and brings that material into the cell. So essentially what the cell is doing with this process is the cell is sampling the extracellular environment. So as such, this process is not very selective, or we describe it as non-selective, because really the cell is just permitting anything that's in the external environment, or I should say a little sample of what's in the external environment, to fill this vesicle, and then the cell brings it in to essentially kind of analyze it and use that information to change what that cell is doing. So this is a pretty common practice for cells of the intestines, where absorption of nutrients from the lumen, or the inner part of the intestine, is important, because right after a big meal, of course, we've got food and other and even waste moving through the intestines. But between meals, generally the intestines might start to empty out a little bit, and so it's important for those cells to kind of constantly monitor the environment inside of the intestines, so that the intestines can release digestive enzymes and juices and things like that to aid in the digestion of those and breakdown of those substances. Last but not least, we have receptor-mediated endocytosis, and this is a mechanism that uses receptors, pretty similar to what we saw with phagocytosis. And indeed, the mechanism here is pretty similar to phagocytosis. So what we see with receptor-mediated endocytosis is that when very specific molecules bind to receptors on the outside of the cell creates a vesicle and brings that material, or that chemical, into the cell. Now, this is particularly useful for concentrating materials that are in really small quantities in the extracellular fluid, and so this could be things like enzymes, hormones, low levels of certain micronutrients such as iron. So this is a really helpful mechanism for selectively kind of plucking those materials out of the extracellular fluid, concentrating them in a vesicle, and bringing it into the cell. We also see, kind of unfortunately, that this is the mechanism, receptor-mediated endocytosis is the mechanism that is hijacked by viruses like the flu as a way to make their way into our cells and make us sick. So this mechanism can also be taken advantage of by viruses and other microorganisms that infect us. So that receptor-mediated endocytosis finishes up our review of the endocytosis mechanisms. Now let's briefly look at exocytosis. So vesicle processes that are ejecting substances from the cell are referred to kind of collectively as exocytosis. And this process is essentially the reverse of endocytosis. So with exocytosis, we see that the vesicles that contain some kind of cargo inside of them, and these vesicles are inside the cell, these vesicles will fuse with the plasma membrane when they're stimulated to do so, and this fusion of the vesicle to that plasma membrane results in the ejection of that material from the vesicle, so that material will then leave the cell. Exocytosis is a pretty common mechanism for releasing things like hormones, neurotransmitters. So these are the hormones we call are the chemical messengers of the endocrine system, and neurotransmitters are the chemical messengers of the nervous system. So this is a pretty common mechanism that we see for releasing chemical messengers into the tissues or into the bloodstream. Exocytosis also moves relatively large proteins, in particular glycoproteins like mucus, and even some cellular wastes and debris are also released from the cell by exocytosis.